Okay, so the last topic is 1.4, the transport of nutrients across the plasma membrane. Um, please remember to review the links that I shared on Google Classroom. It will, I believe the animation, the visuals will be the best way to study this chapter. But anyway, let me just walk you through the concepts that you need to know here. There's three major types of transport. You have bulk transport, you have passive transport, then you have active transport. So let's start from the bulk transport. So bulk means a large amount. So how do you transport large amounts of solute? For example, a big bacteria, how do you transport it inside of the cell? Or how do you transport large amounts of wastes outside to be excreted from the cell? Or how do you transport large amounts of antibodies that will be needed to that is needed to be secreted by the cell? So we're looking at how big how large molecules, large substances, large amounts of waste are transported in and out of the cell. So we have two processes. We call it endocytosis and exocytosis. So endo from the word inside, exo from the word exit. So endocytosis is how to move the materials inside the cytoplasm or inside the cell. So by the way, these are your answers from today's class. So you said in endocytosis, materials are taken inside or inward the cell. And examples are nutrients, or this dot, or nutrients that are dissolved in aqueous solution. So even some water is engulfed inside the cell. The opposite is your exocytosis. So exocytosis, you're releasing useful substances. That's why it's a positive right there, such as digestive and enzymes. Or you're excreting waste products. That's why there's a negative here. So you're releasing useful substances. That's called secretion. And when you're releasing harmful substances, that's called excretion. All right. So towards the inside is endocytosis, to the outside is exocytosis. And how does the cell do this? So the cell needs to form vesicles. Again, review the animation. So when the cell undergoes endocytosis, a portion of the membrane pinches inward to form a vesicle. And that vesicle carries the nutrients, and it can be distributed inside the cell where it needs to be used. The opposite is when vesicles are formed from the Golgi apparatus, and then it merges to the plasma membrane and becomes part of the plasma membrane, simultaneously releasing its contents outside of the cell. So vesicles are important. They are a sac of membrane, so they actually look like the plasma membrane with proteins embedded on it. And these vesicles transports all of these nutrients and waste products into the different parts of the cell, so from one place to another. Now we have passive transport. There's three kinds of passive transport: simple diffusion, facilitated diffusion, and a special type of transport for water molecules called osmosis. So all of these transport mechanism does not require energy. That's why it's X right here. And this is where substances move from high concentration to low concentration. So I can rephrase that by saying substances move along the concentration gradient. So from high to low concentration, it automatically happens even without supply of energy. Now, let's talk about simple diffusion. What can diffuse easily through the membrane? So small nonpolar molecules such as CO2, oxygen, and some water. So they're very small and they're nonpolar. So even if the hydrophilic phosphate heads are even if the phosphate heads are hydrophilic, and even if these are nonpolar, they can still pass through the membrane because they're very small. However, larger molecules such as your glucose, such as your amino acids, and minerals or ions like potassium and sodium, they have a charge and they're large, so they cannot simply diffuse through the plasma membrane, and they have to use some form of help from proteins. That's why facilitated help. So they can move across the membrane through channels or carriers. So these are proteins found in the plasma membrane where your charged particles can pass through. These channels are specific. So glucose channel can only allow the movement of glucose. Potassium channel can only allow the movement of potassium. Okay, and then we have the special case for the movement of water we call osmosis. Now, just a review, if the solute concentration of the inside of the cell is the same as the solute concentration to the outside of the cell, then the cell is said to be in an isotonic solution. If my cell is placed in a solution with a lot of solute concentration, then the cell is said to be in a hypertonic solution. That's what's going to happen to the cell if it's put in a hypertonic solution. All of the water will go out of the cell and it will diffuse out of the cell. Therefore, the cells will shrink. How about hypotonic solution? Hypo means less. So if you put your cell in a solution with less solute concentration, the water from the outside of the cell will move into the cell, and the cell will slowly and eventually swell and burst. So how is this relevant in biology? Now, different cells will have their different isotonic solution, different isotonic concentrations. So if we harvest organs, let's say the kidney or the liver or the heart for organ transplant, we have to store them in isotonic solutions. If we put your organ in a hypertonic solution, like a solution with large amounts of minerals and solutes, what will happen to our organ? Of course, the organ will shrink or burst. What do you think? Let's put in a hypertonic solution. So the answer is the water from your organ will diffuse out and will go into the outside solution. Therefore, your organ will shrink. Now, if it's a hypotonic solution, meaning there's more solute concentration inside of the cell, if you put it in a hypotonic solution, the water from the outside will move to the inside of the cell and your cell will swell and burst. So again, different organs, different tissues will have their own isotonic concentration. If it's put in an isotonic concentration, we expect that nothing will happen to your cell. It's neither going to shrink nor burst. All right, then active transport. 3 important things you need to know to describe an active transport. So, contrary to passive transport, active transport transports your substances against the concentration gradient. So instead of from high to low concentration, your substances will move from low to high concentration. How is this possible? By using a pump protein. So pump proteins needs ATP to force or to pump your substances from low to high concentration. Again, please review here the sodium-potassium pump. That's an example of an active transport that you should be able to discuss. Okay? So, after listening to this video, please make sure to watch the animation of each of these transport mechanism across the membrane. Okay, let me end with showing you all of the transport different ways to transport nutrients in one picture. So you have here diffusion, as you can see. You have a high concentration of these substances, and it just easily diffuses. It passes through the plasma membrane from high concentration to low concentration. Examples are oxygen and carbon dioxide. You'll have here facilitated diffusion. So as you can see, this is still from high concentration to low concentration. However, for these substances, they could be either very large or they are charged. Therefore, they cannot pass through, they cannot penetrate the phospholipid layer. Instead, they use their specific channels or carriers to move the substances from one side of the cell to the inside of the cell. You have active transport. It uses ATP, so attachment of the ATP changes the shape of this protein. Thereby, even if there's many of these diamond-shaped substances here, you can still force your substance to move from low concentration to high concentration. Then you have endocytosis and exocytosis. So it's not here. Think vesicle transport for exocytosis and endocytosis.